Hello everyone, I'm your TA Misan Nyembra, and today we're going to be looking at equine genetic disorders. Now, in order to understand this lecture fully, I would like to familiarize or refresh our memories of some common terms used in the study of genetics and other uncommon ones that you would come across in this lecture. Now, let's start with our building block, which is the DNA. DNA, which stands for deoxyribonucleic acid, is the molecule containing all the genetic instructions needed for an organism to fully function. It is comprised of bases or nucleotides, a sugar phosphate backbone, and hydrogen bonds, as you can see in the picture in the top right. Now, a gene is a unit of heredity or a segment of DNA. They are responsible for observable characteristics that we see in a horse, like its eye color, coat color, and even its behavior. Because of this, it is important to understand reproduction from a genetic perspective and not just a breeding perspective, like we have seen this semester. Now, these long molecules of DNA that contain the genes are tightly coiled and organized into structures called chromosomes. A horse gets 2 copies of each chromosome, one from the mother and the other from the father, just like humans do. But unlike humans that have 46 chromosomes, horses have 64 chromosomes. 62 of these chromosomes are autosomal chromosomes or autosomes, while the last 2 are sex chromosomes. Here's an example of the human chromosome categorized under autosomes and sex chromosomes. And to the right of that, we have a horse's karyotype showing all 32 pairs of chromosomes. Horses are referred to as diploid because their cells carry 2 copies of each chromosome, just like humans do. But notice the absence of a Y chromosome, telling us that this picture here shows a female horse's karyotype. We can think of the egg and sperm cell as the sources of heredity because they pass on the genes to the offspring and explain how traits are carried from one generation to another. However, variation is also bound to occur during formation, and this can sometimes result in a mutation. Now, I know this might be a bit confusing or it might be like a workover depending on your background, but just try to understand how the different pieces fit together. Now, a mutation is a permanent change in the DNA sequence that is either as a result of environmental factors or replication errors. There are several ways in which mutations occur that would be discussed further on in this lecture. But for now, we are going to talk about different forms of a gene that are referred to as alleles. For our lecture, we would classify genes as either normal or mutant. An individual is referred to as homozygous when it is carrying 2 identical alleles for a particular gene from both parents, and heterozygous when it is carrying 2 different alleles. Genetic conditions can be classified as either inherited or spontaneous. The inherited disorders, as the name suggests, are passed on from parents to offsprings. Depending on the type of condition, either one of the parents or both are required to pass on this mutated gene. Now that we have defined alleles, we can understand this better by classifying mutations as either recessive or dominant. For recessive mutations, both alleles of a particular gene, of course, have to be mutant for the organism to be affected. That is, for you to actually observe the mutant phenotype, the individual must be homozygous for the mutant allele. Individuals that are heterozygous for the allele would only be referred to as carriers, so you will not actually observe the mutation, but they have the ability to still pass it on to their offspring. An example of a recessive mutation is lavender foal syndrome, and this is common to Egyptian Arabian breeds. Their foals tend to have complications at birth and cannot stand. Their coat color is usually a pale pink, silver, or lavender, as you can see in the picture. Now we're going to watch a short video of the lavender foal syndrome showing how they have complications standing and their coat color. This clip might be very disturbing and difficult to watch indeed, but I want you to see how the foal really struggles to try standing. It sort of looks like it is having a seizure, but really it is suffering from tetany, which is a condition where it tries to really extend its limbs, neck, and back. It is caused by some neurological disorders, which I think you guys would find interesting reading up on. Another example is dwarfism. Foals that are affected are born extremely small compared to their average breed size. There are some variations of this, which I suggest you look up at a later time. But in this picture, you can see that the horse is significantly smaller than the normal horse. Now, others include glycogen branching enzyme deficiency, hereditary equine regional dermal asthenia, it's a tongue twister, and junctional epidermolysis bullosa. Now, I certainly cannot discuss all of them in this lecture, but I do suggest that you look them up at a later time too. Dominant mutations, on the other hand, are observed in organisms that are heterozygous. They only need to have one mutant allele for it to be observed. But let's not forget that they can also be homozygous, carrying two identical alleles of the mutant gene. One example of this is polysaccharide storage myopathy. Horses with this condition tend to overproduce glycogen, so they store excess sugar that is not easily accessed. This causes several symptoms like muscle pain, weakness, and sweating. Another example is malignant hyperthermia. This is a muscle disorder caused by excess calcium release. Affected horses may experience abnormal heart rates, high blood pressure, and muscle damage. Now, going back to the bigger picture, I mentioned that we have inherited and spontaneous mutations. We have just discussed inherited mutations, so let's shed some light on spontaneous mutations. Spontaneous mutations refer to two types of mutation that I'm going to talk about later on in the lecture, which are point mutations and deletion mutations. They often occur during DNA replication and might be influenced by mutagens such as radiation, like X-rays and UV rays, heat, or chemicals. Research has shown that you can find spontaneous mutations mostly in female horses where they have an abnormal number of sex chromosomes, usually one X chromosome instead of two. So referring back to the image of the female horse's karyotype, a mare with a 63X karyotype, which is a mutation, will have only one X chromosome. This abnormality prevents the mare's reproductive tract from developing and functioning properly, though it is impossible to tell just by looking at the horse. Now, lastly, we have mutations that are believed to be as a result of multiple gene abnormalities. Now, I know this was not mentioned initially, but some examples include osteochondrosis, a disease that affects the joints of horses, and roaring, a larynx dysfunction that affects a horse's ability to breathe.